I know. So, I mean, other than. Yeah, but.
Good morning, everybody. People. Something going on that's not aware. Okay, a couple things. First, we will not meet for class on Friday because I will be at the Murdoch meeting on Friday. Anybody else in here going to the Murdoch meeting? Down here for that because they really limited the number of spots that we got this year. So, okay. Now anyway, I will be there. I will be leaving shortly after surgery on Thursday evening, and I have obligations there all day Friday. So my plan right now is I will video a lecture that you guys can watch for Friday so that we don't get behind. And on Thursday, we start our last round of surgery. Isn't that amazing? I mean, it seems like that just flown by, but the guys have all become such experts at it. Guys actually done really well on surgeries this year. I've been pretty amazed at how, you know, I mean, you guys are probably exhausted when you get done, but I remember years where I was here until, you know, very late in the afternoon, and it was a long day, but you know, it hasn't been too bad this year. So you guys done a really good job. Just pat yourselves on the back. So we will we start our urinary surgery this week. The thing about the urinary surgery, okay, and I'm gonna since I have all but 4 of you here now, I don't know where the others are. I'm going to give you a little heads up on some things that you need to look for in that surgery this week, because finding the ureters tends to be a challenge sometimes. This is going to be another surgery where we're going to be making an abdominal incision, so you're going to have to shave down there. It's going to have to be a little lower than for this week. Because we need to get down to the urinary bladder, which is going to be down towards the lower end of the abdomen, but it will be just under the surface. So the bladder itself will be easy to find. Sometimes you have to rotate. Sometimes you have to rotate the urinary bladder to find the ureters because they both come in on one side of the bladder. And so I'm going to even try to draw you a picture here, I think. So, you know, if this is our rabbit's abdomen, you're gonna open it up, and the urinary bladder is gonna be sitting right here. And when you're looking at it, you may not see it. Like I say, you may have to rotate it, because when you rotate it, what you're gonna see is the urinary bladders are gonna be kind of coming off like so on one side. And even then, sometimes they're a little hard to look at. But if you're having issue identifying them, you just you know, tap me on the shoulder, or tap Audrey or Tiffany on the shoulder, and they can help you find. Once you find them, and this is a cannulation that is that actually I think is the hardest cannulation to do. Not because the vessel is like excessively small. We actually use our smallest cannulating tube for this, but because the vessel, unlike a blood vessel, it's made of soft tissue. And so one of the things that can happen as you're putting the cannula into the ureter is that you can scrape some endothelial cells, and that can clog up the cannula, and that sometimes is a little challenging to work with. But you know, we'll do the best we can to make it work, and and get you through the experiments this week and next week. And then, you know, we get to go to Thanksgiving and all say, lab is over, you know, you can breathe a little bit more. Okay. So anyway, you know, that's kind of that's kind of what you're going to be looking for when you're looking for the ureters during the surgery. Another thing that we're going to want to remember to do, so I want everybody to burn this into your brains, is we always take the ureteral cannula and we will fill it with saline before you do your cannulation. And then we will take a hemostat and we will clip one end of it to seal it. And then you will cannulate. Once you get the cannula into the ureter, then we will open up the ureteral cannula. And the reason we do this is to save you guys all kinds of time sitting around the table waiting for that urine to meander its way down the cannula. This way, if we fill it with saline, once you get the cannula done and the urine is being produced, you can start the experiment right away. Believe me, it saves a ton of time. The rate of urine production differs with every rabbit. Okay, so some of them are just produce copious amounts of urine, sometimes not. So anyway, may you all get rabbits that produce copious amounts of urine, so that always makes it. The other thing is that urine production is visually measured, so we count the number of drops. I'm pretty certain that we used to have some odd instrument. It's just like this impedance thing that they used to do, that they used to have in our instructions for measuring peristaltic waves in the small intestine. I've never even seen the device we used to have in the instructions. And finally, after a whole bunch of years, we took it out. I don't know why it took me so long, but there used to be some other device that the drops would drip on and automatically count for you, but we've never used those either. And so it's just, you sit there and you visually count it. So, and the way you'll do that is you'll take the ureter cannula and you'll hang it over the end of the table and let it drop into your trash can. And then just, you know, 1, 2, 3 for the amount of time that you've got to measure for. So anyway, those are little bits of information that will be helpful for you when you get ready to start your surgeries this week. Okay. All right. So for those of you who came in late, just an FYI, we will not meet for class on Friday. I will be at the Murdoch meeting and be busy. They've got me judging oral talks and 8 posters and all kinds of stuff. And it's more judging than I've ever had to do when I've done these meetings. I always have to, all the faculty that go have to judge, but usually, you know, I got like 4 posters I got to judge or something. But since they've really cut the number of people they're allowing to attend this year, they've decided that they can triple the amount of judging or quadruple the amount of judging that each faculty member has to do. But it's Murdoch and you got to do what they say. So that's fine. All right, is there anything else that I got? I will get started grading exams as soon as I can. I don't mean to sound like I'm whining, but you know, because this is all self-inflicted, so I have nobody else to blame but yours truly. I have a lot of grading things on my plate, and some that have been waiting around for a long period of time. As I've hit things that were a little higher priority, like RAP reports and things. So I may not get started on the exams for a couple of days. So forgive me for that, but I'll get to them as soon as I can. Okay. Do you have any questions before I get started? Yes.
So all this content that we're learning will be on during the finals week, correct?
Yep. Yep, from this point forward, however far we get. My hope is to get at least through muscles. So So just in case you guys want to put it on your calendar, on December 2nd, I will also be out of town. And so we will not meet on that Friday either. But as with this Friday, you will likely get some sort of a video lecture so that we don't get behind. Like I said, I really want to get through muscles because I think muscles are kind of cool. And, you know, if we don't get to the metabolism chapter, that's fine. Or topic, that's fine. But because that's just kind of an extra if we have time. But I really like to get through muscles. Okay, anything else? I hope everybody had a good weekend. My wife and I spent all day yesterday in Hood River. So we took a little us day. We go there every year around her birthday. Her birthday's in October. And spent a long day there. Got back sometime between 8 and 9 last night, which always makes this morning a little challenging, but you know what? That's self-inflicted too. So, all right, let's get cracking. Okie dokie. So one more little bit of comparative info, and then before we move on to our focus on the mammalian respiratory system, talk for just a couple minutes about birds. And birds are really fun contrast to mammals because of the other endothermic group. But their respiratory system is designed so much differently than what you see in mammals. That the contrast I think is worth doing. So birds being a direct descendant of reptiles. In fact, in case you weren't aware, they, as of a couple of years ago, they now actually classify birds as reptiles. If you didn't know that, okay? They are warm-blooded reptiles and considered to be the closest relatives to dinosaurs. Okay. I know that Nature Day is now that you know that, but it's the truth. But anyway, the avian respiratory system is really interesting because they have a lung here. Turn my thing on. Okay, but this lung design reminds me a lot of a car radiator. And I'll explain why. Okay, but associated with the lungs are a series of air sacs, both anterior and posterior to the lung, that primarily act as holding areas for air as the air is moving through the avian respiratory system. And we'll look at how those work in just a second. The lung itself, a couple of key components. You know, you have the trachea, in which the air comes in. The air will then enter into what here is called the dorsal bronchus. I'll actually give you probably a more modern name for that in a moment. The air will then travel through these small tubes that are called parabronchi. And the parabronchi are the homologue to the mammalian alveoli. So it's where gas exchange actually takes place in the avian lung. The air will then go into the ventral bronchus, and then it will exit the respiratory system. That's a simplistic overview of what happens, and I'll give you a more detailed version of this in the next slide. But the key thing that you should note from this is that the air travels in only one direction through the lung. Okay, so unlike a mammalian lung where air kind of goes in and out, it's unidirectional flow through the avian lung. And this unidirectional flow adds to the efficiency of the avian lung. And the avian lung is probably, well, it actually is the most efficient respiratory system of any bird, short of the fish that we looked at last time. But, you know, a fish-like respiratory system really would not work in a terrestrial animal because gills would work for us. So anyway, it's really quite an interesting design. Now, the other thing about the avian respiratory system that's different than mammals is all the adaptations really are associated with the lung itself. You know, where when you look at mammals, one of the huge adaptations is in the design of our red blood cells. But if you look at birds, their red blood cells are very reptilian. Okay, they're larger, they're nucleated, they have all the organelles you'd expect in a cell. And so in terms of how adapted they likely are not as efficient as a mammalian red blood cell would be. So the big point that I think I want to make with all this is there are different ways to attack respiratory efficiency in terms of design. All right, so here again is just a blow up of a small section of the lung. We have the dorsal bronchus coming, where air is coming in, it will drop down through the parabronchus here. Gas exchange will occur here. You'll see that you have capillaries that are going to be surrounding the parabronchus. Then the air will drop down here into the ventral bronchus where it will exit the lung. Here is a scanning electron micrograph of an avian lung, just so you can kind of get a real life view of it. This is a parabronchus here, this is a parabronchus here, this is a large blood vessel right here, and then the whole structure here is inundated with capillaries. All right, so what makes the avian lung tick? So again, we have air that's coming in through the dorsal bronchus, and then it drops into the parabronchus here, and then we have blood that's going to flow across this. And this is the key to its efficiency. We don't have countercurrent flow like we see in the fish gill, but we do have what's called cross-current flow. And this cross-current flow does increase the efficiency of the lung. So if we look at this diagram right here, we see airflow that's going down through the parabronchus right here. And then we see blood flow that's flowing across the airflow right here. And what we see is we get some oxygen absorption from the air pretty consistently across the entire parabronchus. It's best at the beginning, where you have the largest partial pressure gradient, but you still have some that is occurring as we get towards the end. And so what we can see here is the partial pressure of blood, or the partial pressure of oxygen in the blood as we move down the parabronchus, it declines because there's less oxygen in the air. But when all is said and done, the partial pressure of oxygen in the blood that is leaving the lung is going to be about 65 millimeters of mercury, which means it is higher than the partial pressure of the air that's leaving the lungs, which means the lung is pretty doggone efficient. And avian lungs can push an efficiency of the values I've seen are up in the neighborhoods of as high as 60% oxygen extraction, which is really pretty doggone good. And when we look at the mammalian lung in a second, we'll see that the mammalian lung isn't anywhere near as good as that. Okay, questions at this point? Okay, so summary of the characteristics of the avian lung. The flow is unidirectional. So if you're not familiar, I use the analogy of a car radiator for the avian lung. So with a car radiator, you know, it kind of sits right in front of your engine, and air flows across these tubes down which the water that's being heated in the engine is flowing to cool it off. Okay, and you got that air that's flowing across the flow of water, and so it's pretty effective at cooling the water as it drops through the radiator. And the water flows to the radiator in one direction. It's unidirectional. So it's kind of the same sort of design. Does that make sense to everybody? Excuse me. Okay, so unidirectional airflow. Blood flows across the airflow, so it's cross-current flow. It's considered to be moderate efficiency, but relative to a lot of vertebrates, it's actually pretty doggone good. Lung PO2, just because it's breathing air, is 105 millimeters of mercury. That will be true of any organism that's breathing air. The arterial PO2 is higher than that of expired air, and as I mentioned a moment ago, that highlights the efficiency of the lung. And this is kind of an interesting point, and I don't know really how accurate it is. This is something that textbooks have said, and it actually may be something that an older version of a textbook said. So it says possibly does not require high oxygen affinity. And so now we're talking about the nature of the hemoglobin, okay, that's in the red blood cells. And one of the things, there's been a fair amount of work that's been done, excuse me. Okay, let's try that again. There's been a fair amount of work that's been done in the past, I guess maybe 10 years or so, on birds at high elevation. And one of the things that they've looked at in these studies is that the level of oxygen efficiency for hemoglobins in birds that are adapted or acclimatized to exist at higher elevation, they tend to have a higher affinity for oxygen. So it's not a one-size-fits-all sort of thing. And so this statement may be maybe too far, maybe a much broader generalization than is really fair to birds, because there's a lot of variability in how high the affinity of oxygen is for it, how high the affinity of their hemoglobin for oxygen is, depending on the environment in which they live. And we'll talk about the importance of that a little bit more when we get towards the end of this topic. All right. Questions? All right. Now let's go highlight what you all have doing respiration in your chests. Okay, the mammalian lung. So, unlike the bird lung, the functional unit are these little structures that we call alveoli, which exist in sort of like these grape-like clusters inside your lung. They are little sacs that when you inhale, they will become filled with air, and these little sacs are surrounded by capillaries. And air moves in, air moves out. The term that they use to describe this is called tidal flow. And this limits the efficiency of the lung. And one of the things that is characteristic of a mammalian lung is that the PO2 of the exhaled air tends to be higher than that of the PO2 in your blood when it leaves the lung, which highlights its inefficiency. Okay. And again, it's just because the air comes into your alveoli, it just kind of, whirls around in there a little bit, and then it gets exhaled. There is not like this constant contact between all the air in the alveoli and your blood. Okay. So that does limit the efficiency. But we're still sitting here all comfortable this morning, and so it works. Some characteristics before we start getting into some of the dirty details. Tidal pool type ventilation, mesh type blood flow around the alveoli, so it's not like a counter current or cross current, it just surrounds the air when it comes into these sacs, and the air just kind of mingles around in there a little bit. The efficiency tends to be low, less than 50%, and in general, substantially less than 15%. I've seen some estimates of the efficiency of human lungs that gets down as low as 15%. And a lot of this probably depends on your particular physiological state as well. That may have a lot to do with it. But even so, it's not very efficient. And again, I think I've mentioned this before, that's why artificial respiration works so good, because the air that you exhale still has a heck of a lot of oxygen. And so, you know, you can deliver that oxygen to somebody else. And like I say, some of you have seen me do that for rats, you know. Nobody else has seemed to be as willing to do that as I am, but it's okay. All right, air entering the alveoli is around 105 millimeters of mercury. Again, that's going to be the case with all air-breathing animals. Blood will require a higher affinity for oxygen, so our hemoglobin tends to be pretty good in terms of attaching to oxygen. And then, of course, our blood cells also are highly adapted to maximize oxygen transport because they're smaller, have more surface area relative to their total volume, and they basically are just sacks of hemoglobin, where that's not the case with any other taxon. Arterial PO2, as I mentioned, is less than that in expired air, which reflects the low efficiency. Okay, questions?
Can you explain again why the arterial PO2 is lower?
So if your arterial PO2 is higher than that air, that means you're sucking a lot of the oxygen out of air. If it's lower, it means you're not getting as much out. Okay, so it's just a matter of simple math. You know, if you know, in a countercurrent or a cross current, the air has so much contact with blood in such a way that you have these favorable gradients all the time. So it just removes the oxygen really, really well. And so that blood is going to have a pretty high PO2. But in the case of mammals, you know, you figure you got that sac, these little alveoli, where you've got air running around there, and they are surrounded by blood vessels, but the air, not all the air is in contact with those blood vessels consistently. And so the proportion of oxygen that's going to be removed will likely be impacted somewhat by your respiration rate. So how fast are you bringing air in and pushing air out of those alveoli? Because the alveoli will inflate with air every time you inhale. That answer your question, Tori? All right. Okay, time for some theory. So the mammalian lung is a negative pressure system. Okay, air flows into the lung because you create a negative pressure environment, and it is based on what we call Boyle's law. And so Boyle's law states that pressure is proportional to the inverse of volume. So what that means is as pressure increases, volume is going to decrease, and vice versa. Now, what does that look like? Well, let's look at a little cartoon here. So we have this very diagrammatic lung right here. And we can see that when we compress, we decrease the volume. Okay, all these things get closer together. They're very, very active, and they're putting pressure on the walls of this space right here. The pressure goes up. When we decompress, in other words, we increase the volume goes up, the pressure goes down. Okay, and this here is just kind of an expression of Boyle's law up here. P1 D1 equals P2 D2. Does that make sense to everybody? All right. And so that's the way your lung works. Every time you inhale, you contract those intercostal muscles, your diaphragm. It pulls on your lung and it increases the volume. Okay. So the lung volume increases, causing a decrease in pressure internally. Air will always flow from a region of high pressure to a region of low pressure. So when you expand your lung, when you inhale, at sea level, okay, you decrease the pressure to about 755 millimeters of mercury. There's the value in kilopascals. And as we know, at sea level, the atmospheric pressure is 760 millimeters of mercury. And so that creates a 5 millimeter mercury pressure differential. As I mentioned, air will always move from high pressure to low pressure. So once you have expanded the volume of your lungs, air is going to move in through your trachea and into your lungs. Okay. Here's just another way of looking at that. This is just a very diagrammatic view of an alveoli here. You inhale, the alveoli will increase in size, creates an alveolar pressure that is less than atmospheric pressure, air will move in. Then when you exhale, you will get a decrease in the size of the alveoli, which means that your alveolar pressure is going to exceed atmospheric pressure and the air will be pushed out of the alveoli. In terms of the flow, here's an equation that will describe the flow here. Flow is equal to the difference between the alveolar pressure and the atmospheric pressure divided by the resistance of the airway. And we'll talk a bit about the importance of the resistance of the airway in just a moment. And so if this is a positive value, air is going to flow into the alveoli. If it's a negative value, air is going to flow out of the alveoli. That makes sense to everybody. All right. So I mentioned resistance. So when the lung expands, there is actually forces that will resist the expansion of the lung. One of those forces is just simply the elasticity of the lung. Okay, it's not, the structure is not hard. When you bring air into it, there is a resistance, just an elastic resistance to the expansion of the alveoli. But a more important form of resistance is due to what we call surface tension. And surface tension is exerted because the alveoli are covered by a really thin layer of water. So this thin layer or film of water, you have all these water molecules that are interacting with each other. They're attracted to each other by hydrogen bonding. And those hydrogen bonds are actually really, really strong. And they provide substantial resistance to the expansion of the alveoli when you inhale. Right? Does that make sense to everybody? So it's almost like all those water molecules are holding hands along the surface of the alveoli and saying, no, you're not going to expand this surface. Now, if the people who study the physics of these things have actually done calculations which suggest that the mammalian lung should not work because of that surface tension. It should actually collapse. So let's talk about how that works. So your lungs overcome this by secreting a substance that's called surfactant. Just put that in the back of your brain right now because I'll explain what surfactants are in a moment. But without this surfactant, which is necessary to overcome this resistance that some predict should cause the lung to collapse, the function of the alveoli is explained by what's called the law of Laplace. And the law of Laplace, basically what it says is the pressure inside the alveoli is equal to 2 times the surface tension divided by the radius of the alveoli. Now, something to know about the alveoli of the lung is that they are not all of uniform size, which tells you that this radius is going to be really important. So if you get a scenario where you have 2 alveoli in the same cluster, one large, one small. So the A here is large, B here is small. And the surface tension for both of these alveoli is the same. The only thing that's different here is the radius. So if we apply this equation right here, in this case where we have no surfactant, nothing to get in the way of the differences between these two alveoli, then what happens here is we end up with a scenario that the pressure in this large alveoli is going to be less than the pressure in this small alveoli. You all following me? I'm looking for spinning heads out there. Okay, since the pressure in this large alveoli is lower than the pressure in this small alveoli, what will happen then is this small alveoli will literally be sucked into that large alveoli. That wouldn't be a good scenario, would it? However, your body deals with this by releasing a very, very important substance that's called a surfactant. And the surfactant has some very important properties that adjust surface tension of alveoli of varying sizes such that the pressures in these alveoli will equalize out. And because the pressures will equalize out, you will not have a case where these small alveoli will then be sucked into these large ones, so the lung does not collapse. So clearly, what these surfactants are doing is they are altering the surface tension in these alveoli that are of various size. Questions about anything I said on this slide? Okay, so what are surfactants? Surfactants are molecules that are very much like phospholipids, and to some degree like a detergent, a soap. And they migrate to the air-water interface on the surface, inner surface of the alveoli. They are amphipathic. So remember I mentioned they were somewhat like phospholipids. They have a hydrophilic head and a hydrophobic tail. And they arrange in a monolayer. So they look like this here. So you have the hydrophilic heads here and the tails kind of sticking up, and then your water layer right here. And what they do is they will interfere with the interaction between water molecules. And essentially what they do is they prevent the water molecules from effectively hydrogen bonding to each other, which minimizes the amount of surface tension that you're going to get inside that alveoli. Does that make sense? Yeah.
Why did you say that they were like a detergent?
Because detergents, one of the things that they will do is they can disrupt hydrogen bonding on surfaces. So I'll give you an example. Maybe some of you even did this in your early school days. There's an experiment where you can take a small pin, a very narrow pin, and you can get it to float on water because of the surface tension. And then if you take a drop of just dishwashing soap and put it in the water, that pin will just sink. Y'all know what a water strider is? If you really want to be mean and nasty to a water strider, you put it in a little container, and then you know how they literally walk on water? Put a drop of dish detergent in there, and that water strider is going to sink because it sits on that water because of surface tension. When you apply an herbicide to plants, often people who do this professionally will put a little bit of dish detergent in with the herbicide to serve as a surfactant because it helps the herbicide to overcome surface tension on the surface of the vegetation, and it helps the herbicide get into the plant. So, did that answer your question? So I know the next day you go see Waterside, everybody's gonna go out and start dumping dish detergent into their water, watching it sink. Other questions? All right, now let's talk about gas diffusion. And I mentioned this last time, but we'll talk about it formally now. The fusion of gases occurs down a pressure gradient as opposed to a concentration gradient. So, time for a brain teaser. You have a container here that's separated by a membrane. On one side, you have olive oil. On the other side, you have saline. Olive oil will dissolve twice the amount of oxygen that can dissolve in saline. So, on the olive oil side, you have a high concentration of oxygen. On the saline side, you have a relatively low concentration of oxygen. Even so, oxygen will always flow from the saline to the olive oil. So it is flowing against the concentration gradient. Why? I have already told you everything you need to know to answer this question.
Is the olive oil pressure gradient lower?
You're getting there. The olive oil itself doesn't have a pressure gradient. I'm going to draw you a couple of pictures here. And I want you to just think in terms of classic diffusion. So if I have a chamber that looks like this, and I've got a membrane right there, and I have a high solute concentration here and a low solute concentration there. Which direction is solute going to flow? Left to right or right to left? Is that left to right or right to left?
The high solute concentration.
Yeah, it's gonna flow in this direction. The size of the container doesn't make any difference. It's the concentration that dictates that. So now I have a low solute concentration here, high solute concentration there. Which way is the solute going to flow? Right to left or left to right? Does the size of the container make a difference? Okay. So which way is it going to flow? Go ahead.
I'm just confused because, so I know that it flows from like a high concentration to a low concentration, but right there it says that fusion of gases occurs.
I'm getting to that. I'm trying to set up to help you understand this a little.
Okay, so in this.
We're just talking about solutes, not gas right now.
Okay, so it's gonna flow from right to left.
It's gonna flow from right to left. Yeah, it's gonna go that direction. Okay. So now we know. Okay. Now, actually, let me hold it. So we know we have one side of this container here that can hold a lot. And one side over here that cannot hold a lot, holds a lot less, but we still have the flow in this direction. What drives the flow? What drives the diffusion of gas? It's not concentration gradient, right? It's what? It's the pressure gradient. So what does that tell you? What does that tell you about the pressure, the pressure of oxygen in the saline cycle? Is it high or is it low? It's high. The pressure over here then has to be low, correct? In order for it to flow in that direction. Now, the reason that I went through this little exercise with you is because this, what I've set up here, is a lot like this right here. This can hold a lot, but it's sort of like this having a much bigger volume over here. I can create a low solute concentration over here, even if I have twice the amount of solute, just simply because I have a bigger container. Does that make sense? And I can create a high solute concentration over here with less solute than I have over here because I have a smaller volume. I don't need as much to make a high concentration. Yeah.
So if you have, like, because the olive oil can dissolve twice as much, that's why it's able to have a lower pressure gradient.
I'm sorry, say that again?
Because the olive oils are able to dissolve twice as much, that's why it has a lower pressure gradient.
Yeah, it's sort of like being a big container. Okay? It's sort of like looking like this. And so the solubility has a lot to do with with what the actual partial pressure of oxygen is going to be in that chamber. Okay. So the olive oil, even though the concentration is higher, the partial pressure is only filling 40 millimeters of mercury. And in saline, even though the concentration is lower, since it has less solubility, the partial pressure is substantially higher at 100 millimeters of mercury. And so it's important to understand that gas moves on the basis of a pressure gradient, not solubility. Any questions about that? Does that make sense to everybody? Good. All right. So I've been throwing around this term partial pressure. What does it mean? Okay, so atmospheric gas. We know that it's sea level, and we're close to sea level here. That the total atmospheric pressure is 760 millimeters of mercury, 101.3 kilopascals. This is the pressure that atmosphere is exerting on the surface of the Earth due to gravity. So that 760 millimeters of mercury, it is the sum of the effects of all the component gases that make up our atmosphere are exerting on the surface of the Earth. And we know that the atmosphere is about 79% nitrogen. That's 106 or 601 millimeters of mercury, or 79.9 kilopascals. 20.95% oxygen, which is 159 millimeters of mercury, or 21.1 kilopascals. 0.03% carbon dioxide. You can tell that this is an old number because it's higher than that now, which is 0.3 millimeters of mercury or 0.04 kilopascals. And then there are some other trace gases. Okay. Each of these pressures that I've listed here that are specific to each of these individual gases represents its partial pressure. So the total pressure of a system is going to be equal to the sum of the partial pressures of all its component gases. Does that make sense to everybody? If it does, then we will call it a day. And I will send you out to have dreams about partial pressure until Wednesday. And we will pick this up here.